Dr. Haynes is willing to provide you with some time to think for that. So one thing we want to encourage you guys to be doing is to be thinking about integration of locations now. Not so much obsessing about, you know, specific details that may be beyond what we've even talked about in lecture, and rather thinking about how do all these parts fit together in functional ways. Okay, and so that's going to involve you thinking through whole systems, whole pathways, drawing them out, and understanding why they go to where they go, what they do at those different locations. And so I think that, you know, if you bring an atlas, bring paper and colored pencils. You know, you can use the cross-sectional sheets that are on the big poster board there, and think through integration of pathways. That's going to be your best bet for how you should be reviewing. I also just want to note to you that in the slides that we have for today, there's a couple of slides about spinocerebellar pathways that are in your in the file I gave to you. As I was going through looking at my slides, I was afraid I was going to run out of time, so I hid them from our discussion there, but they're in your in the file I gave to you guys. So hopefully you were looking at the mystery pathology. And so we have a 37-year-old woman who's having a number of issues of a fairly short duration. It's only been about a week that she's had these issues. And you can see that they represent a number of different types of functions or tasks, but importantly, there's a differentiation between what is not functional and what is, right? Pinprick and temperature are intact, but she's got all these other issues going on. Okay, so keep this in mind as we go through class today. What's causing her to have this paresthesia, these sensations that aren't real, the clumsiness, the problems with dexterity that she's experiencing. Okay, so just to go back to our sort of oversimplification of sensory systems in general, I want to apply that now to somatosensory systems. And recognize that the reality that we're talking about for somatosensory systems is external locations, that is on the body, and internally on the surfaces of the body, the structures inside, so visceral as well. And we're talking about the body and the head. Okay? And in terms of receptors and sensory transduction processes, some of the things that we are going to be able to distinguish are going to be discriminative touch, vibration, proprioceptive input, crude touch, sort of pressure kinds of things, as well as temperature and pain. And ultimately, that kind of information is going to get propagated up through pathways like the posterior column medial meniscus, through the trigeminal thalamic, cerebellar pathways, and the anterolateral system. And so for today, we are just focusing in on the discriminative touch, vibration, that aspect of somatosensation, as well as proprioception. And so the parts about pain and temperature are going to be on Monday. Okay, so when we talk about somatosensation, we're focusing in on those tactile, vibratory, proprioceptive inputs. We need to start with the receptors that detect those types of stimuli. And so they can be broadly categorized as being either rapidly adapting or slowly adapting. Okay? And so if they are rapidly adapting, what that means is that they respond to the initial onset of a stimulus. When something is initially applied, they fire off a bunch. But if that stimulus is maintained, they stop firing. But when the stimulus is removed, they're going to fire again. Okay, so they respond to the changes in that. And the types of fibers that they're associated with are A-beta fibers. And so these are fibers that are fairly large in diameter and rapidly conducting. So examples here from the picture, Meissner's corpuscle, so here's a drawing of that. And they are cutaneous, they're going to respond to more of light touch. Hair follicles, down here, are particularly good in detecting direction and motion of the stimuli. And then Pacinian corpuscles down here. Notice that these are very close to the surface, right? And this is much deeper down. And they are good at detecting vibrational stimuli. And so slowly adapting receptors are receptors that are going to continue to respond throughout a sustained presence of the stimulus. So they'll start responding when the stimulus is applied, and they'll maintain the steady rate of response during the entire duration of the application of the stimulus. And so they're particularly good then at telling you about duration of the stimulus, about the intensity of it, those kind of details. Same kind of fibers they're associated with. So A beta, so again, more rapidly conducting fibers, large diameter. And the types of peripheral receptors that are slowly adapting are Merkel cells, that's here. And they are, again, those are more superficially located, right, more cutaneous, good at pressure. You have hair follicles. Again, so notice I have hair follicles in the rapidly adapting category. Hair follicles can behave differently there. So hair follicles are here again. And then the Ruffini corpuscle. So that's a deep location receptor that is particularly good at recognizing stretch. I want to point out Meissner's corpuscles, those are the rapidly adapting, and Merkel cells are the slow adapting. Those are the ones here and here that are cutaneous. Right? They're right there at the basal parts of the epidermis, or the most superficial parts of the dermis. They have really small receptive fields, and you find them in high concentrations, like in the fingertips. Okay, so in this picture, you've got how many of these receptors do you have per unit of space, and the degree of spatial resolution that you get out of those areas. You can see where you have high concentrations of these receptors, like in the fingertips, you have excellent spatial resolution. So you can detect very, very small things. Using your 2-point discrimination is very good in the fingertips, right? And not so good here in the palm of your hand. You know that anyway. You never use the palm of your hand to try to tell the detail of something you were touching, right? General pattern is that you have smaller receptive fields in areas like here on the fingertips, small receptive fields that have a high density of afferent innervation. So you see lots more fibers coming in to smaller areas. And that corresponds with having greater discriminatory abilities and disproportionately larger map areas. So you can look at like the fingertips area in the hand, and you see on the map how much larger that area of space is in cortex. Conversely, your back, you have terrible localization ability on your back, right? And you can see how large that receptive field is, right? And that corresponds to your whole back has less cortical territory than your fingers, your individual fingers. Okay, so I also need to mention to you the types of proprioceptors that are located in the periphery. And you tend to find those in, you find them in muscles and tendons, you find them in some connective tissue, and the joint capsule space. So I want to mention two types, muscle spindle receptors and Golgi tendon organs. Okay, so muscle spindle receptors are particularly good at telling you about the length of the muscle. Okay, if the muscle is being stretched or contracted. And so muscle spindle receptors are long, thin stretch receptors that you're going to find within a skeletal muscle. So this figure in your book, I've just blown up one little bit of it, but if you could see the part that was up here, you'd see that the muscle spindle receptor is contained within the bulk of the normal skeletal muscle fibers. Okay? And so within the muscle spindle receptor, there are two types of fibers. Okay? There are nuclear bag fibers. That's this guy that's kind of fatter in the middle. And then there are nuclear chain fibers. This guy is sort of skinny and looks a little bit more like a banana in that drawing to me. So sort of, the reason is it's about the position of the nuclei. Okay, in the chain fibers, the nuclei are in a straight line, single file organization, and in the bag fibers, they're more adjacent to each other, so it forces it to be wider in the middle. Okay, so they have this central area that doesn't have the typical microfilaments involved in contraction, and they get a sensory innervation in that area, okay, and then they do have some contractile elements in the periphery. In this central area, they get afferent innervation from endings, so they're called primary endings, so they're type 1A fibers. So those others I was telling you about before were type 2s. Type 1s have an even larger diameter, they're even faster in their conduction. Okay? And so the type 1s here are really good at the onset of the stretch. The type 2, the secondary endings, they innervate out here at the periphery of these interfusal fibers, and they are going to be telling you about the sustained firing during the stretch. Okay, and then Golgi tendon organs. So muscle spindles are really good at telling you about the stretch of a muscle. Golgi tendon organs are particularly good at telling you about the tension or the force. And so right here you have the Golgi tendon organ. All right, so here's the muscle, and here's the tendon that's connecting it. And so you find the Golgi tendon organs sort of in this interface area, and they get activated when I put it in quotes, kind of when they get squeezed by the surrounding connective tissue in the tendon when the muscle contracts and pulls on the tendon, and so it kind of squeezes in then on the Golgi tendon organ, and that activates it. And these guys are particularly good at telling you about limb position, joint position, and they can also give you dynamic limb movement information. They are innervated by type 1 fibers as well. So remember those type 1 fibers that went to the center of the muscle spindles, and then you've got another type of type 1 fiber that's coming to the Golgi tendon organs, carrying that information away to the central nervous system. So type 1, so they're going to be really most rapidly conducting as well. Okay, so we've identified what kind of receptors are located in the periphery to tell us these somatosensory stimuli. Now, how do we convey that information up through the central nervous system? Okay, so this slide is a sort of a summary of all the steps in the pathway. Okay, the basic idea is that out of the periphery are the receptors. I told you about what kind of afferents innervate them in terms of axons. Then, where are the cell bodies, right? So the cell bodies here and here are in the posterior root ganglion. Then those axons come into the posterior column, and they ascend in the fasciculate gracilis or cuneatus. Second order neuron is located in the ipsilateral posterior column nuclei. Those cells have axons that cross and ascend in the contralateral medial meniscus to go to the contralateral VPL of thalamus, so that's up here. Those guys send axons through the posterior limb of the internal capsule up to postcentral gyrus and posterior paracentral gyrus. So that's the whole thing in one slide. Okay, but we're going to break that down and go through that step by step now. Okay, so those large diameter, yeah.
It's about the previous slide. I don't understand what's the difference between one side and the other. One is the line.
These are the cell bodies where the neurons are located. This is where I put the axons. So it's sort of here and here, here. Just zigzag your way up the slide. Okay, so those large diameter afferents that I was talking about, those type 1s, type 2s, are going to be, so there's the posterior column, or the posterior root ganglion, so there's the cell body. Those afferents are coming in, and what you should see is that they're coming in just medial here to the dorsolateral tract, right? So they're just medial to where that sulcus is, and they enter in here at this medial root entry zone, and then they are going to turn and ascend. Some branch off, and this participates in spinal reflexes. You can see here, this is a picture of the lumbar cord, and if we look at this sort of close-up view here of the posterior root, you can see these fibers coming in, follow them along right in through your airway, right? So they're coming in just medial to that dorsolateral tract. And then they're going to turn and they're going to ascend. Now I want you to note that when they come in, they're arranging themselves in a somatotopic way. Okay, so that the sacral, fibers representing sacral areas are most medial. So I put an S here near the posterior median sulcus. So it's most sacral here, and then we move lateral toward the uppermost lumbar areas, moving laterally in the psoas fasciculus. Okay, so again, I think I've told some of you guys, I think about this like an elevator in a tall building, and you get it on the bottom floor, and then when you stop the next floor up, you got to scoot back, right, to let more people in, and you keep scooting back. And so that by the time you get to the top, those people who got in first are in the back of the elevator, right? Same sort of idea here. You get pushed medial, and then stack them up, moving laterally. So you're going to ascend through that posterior column, and now I'm showing you a picture of cervical cord. Okay, and so now, not only do we have the gracile fasciculus, right, but now we've added the cuneate fasciculus as well. And so the gracile is I sort of put a blue area here, and then the pink for the cuneate. So the posterior intermediate sulcus is the dividing point between these two. You can see then in the drawing here, we've got our lower extremities represented in the gracile. And remember, so this is most sacral, moving upward. At about T6 is where we transition from gracile to cuneate. And then we'll continue moving, so then this is going to be thoracic, moving upward toward the most upper levels of cervical and neck area, right? So you have that somatotopic pattern all the way through the posterior column. For the posterior column, the blood supply is coming from the posterior spinal artery, right? So you remember the posterior spinal artery shown there basically sits in this area near the posterior lateral sulcus. And so you've got one here and you've got one here on this side. And so you can see then that they are supplying the territory of the posterior column. So I told you that there's a little bit about those spinocerebellar tracts, and so that's the blood supply to them because they're located very lateral here and here. That blood supply is coming from the vasa corona, what's wrapping around the cord. You've got the anterior spinal artery. Remember that guy that's doing all this area. So it's going to get your lateral corpus tract. That's going to get your anterolateral system. And then the posterior column getting the posterior spinal artery. Okay, so we're ascending up the posterior column. And remember, these axons are still the axons from the first order neurons that are out there in the posterior root ganglia in many cases. Okay. They are heading toward their second order neurons in the pathway, which are found in the posterior column nuclei. Right? So the nucleus gracilis, the nucleus cuneatus. Okay, so they're ascending up here and here, ipsilaterally heading for their second order neurons located here. Okay? And you can see it here as well, right? So in caudal medulla is where they find those second order neurons. So the crossing happens with the axons of the second order neurons. And so that's where we're going to cross from the ipsilateral to the contralateral side. Okay, so we've got those second order neuron cell bodies in the nucleus gracilis and nucleus cuneatus. Okay, so this view is showing you at the level of the motor decussation. All right, what you remember is most caudal part of medulla, right there at that spinomedullary junction area, right? And so there you have the motor decussation. We have nucleus gracilis, and nucleus cuneatus. And you still can see a lot of the fasciculus cuneatus in this area as well. Okay, so then, and that's at this level. Then we go a little rostral, and we get this view. Okay, and so now we still have the posterior column nuclei, or we don't have that big bit of the fiber tract anymore. But now we're at the level of the sensory decussation. So that is where these guys send off their axons to go join up the medial lemniscus. Okay? And so those axons, can you kind of see these little like stripes cutting through there? Kind of banking around. That's the sensory decussation. Okay? Those are those axons heading to the contralateral medial lemniscus. Okay? Okay, so I tried to kind of trace it for you here so that you can see it. So we've got our lower extremities in the gracile and our upper extremities in the cuneatus. Lower extremities are going here to the more anterior parts of the medial lemniscus, right? And your upper extremities are going to the more posterior parts of the medial lemniscus. And so we maintain the somatotopic pattern that we had as we came up through the posterior column. Okay, and so then these internal arcuate fibers are where we have our decussation. So now we've crossed over, and now we have the medial lemniscus oriented in this sort of anterior to posterior orientation. And you can see then that continues up to the rostral medulla. So I have the medial lemniscus here, with the lower extremities more anterior and the upper extremities in a more posterior position. In the medulla, the posterior column, as it reaches the medulla, and the posterior column nuclei are supplied by the posterior spinal artery. Okay, so that's here. You can see that through there. The medial lemniscus is supplied, so we start seeing it here, is supplied by the anterior spinal artery in these levels of the medulla. As we start to transition up into pons, that blood supply changes from medial lemniscus. Okay, so the medial lemniscus is going to change its location and relative orientation as it ascends through the brainstem. Okay, so it's going to start off in this sort of anterior posterior orientation that I was showing you. Here it is, right? Anterior to posterior orientation. And it stays that way through the medulla. Okay? But when we get to the pons, it makes an abrupt change, right? It's shifting itself. So you can see that here at this mid-pons level. Now, the medial lemniscus is running in a more medial or lateral kind of way. Okay? And in terms of maintaining the somatotopy, that maintains itself, and I think of it like sort of like it's pivoting. So the upper extremity kind of stays where it is, and the lower extremity pivots laterally. Okay, so we still have the somatotopic pattern, but now the lower extremities are more laterally represented, and the upper extremities are more medially represented. Okay, and then as we continue, we get up into the midbrain, and that pivot of the lower extremities continues. So now the medial lemniscus is even turned so that the lower extremities are moving more dorsal compared to the upper extremities. And so you can see that illustrated here, and then flip that over in your mind to see it over here in the clinical orientation. Okay, so in the level of the pons in the midbrain, the blood supply for the medial lemniscus has gone from anterior spinal artery, which is the medulla, to branches of the basilar artery. So here's the medial lemniscus, and so you got these branches, the paramedian branches of the basilar, and as you get up into the midbrain, where you get medial lemniscus, then you start getting branches of the posterior cerebral when you get to the most rostral parts of the midbrain. So the medial lemniscus has traveled all the way up through the brainstem, and it's going to terminate in the vitreoposterior lateral nucleus of the thalamus. Okay, so that is shown in the drawing right here. So medial meniscus is ascending up through and terminating here. And see the somatotopic pattern maintains itself here, so that the lower extremities are more laterally positioned and the upper extremities are more medially positioned. And you can see that illustrated here as well. At the level of thalamus, the blood supply is now coming off of the thalamo-geniculate arteries, which are also supplied by posterior cerebrum, but a different branch. And I wanted you to get a chance to see what VPL looks like in a couple of different views and what's surrounding it. So ventral posterior lateral nucleus, so I've got a coronal view and a axial view. You can see VPL traced it out for you here, and then traced it out over here. So you've got pulvinar around it, you've got the centromedian nucleus is near it, the internal capsule is on the lateral side of it, and the ventral posterior medial nucleus that we'll talk about in a moment is just medial here. Okay, so that's where it's positioned there in the diencephalon. Those projections go in the posterior limb of the internal capsule up to the primary somatosensory cortex. And you remember that that's located in the postcentral gyrus, and then on the medial surface, that's on the posterior paracentral gyrus. And so you can see that map here, right? So we have our most our upper extremities and face in this lateral area, and our lower extremities here on that medial surface. The postcentral gyrus, you kind of see it here. We've got primary somatosensory cortex being Brodmann's areas 3, 1, and 2. Right? So you've got these two subdivisions of 3 and 1 and 2 making up primary somatosensory cortex. In terms of blood supply, now that we're up in the cortex, the blood supply changes depending upon whether you're talking about the upper extremities or the lower extremities. So if you're talking about these lower extremities that are on that medial surface in the posterior paracentral gyrus, then the blood supply is the anterior cerebral artery. Right? But if you're talking about the upper extremities of the face, then the blood supply is coming from the middle cerebral artery. Okay, so now we have taken proprioceptive, tactile, vibration kind of information all the way up through the pathway to cortex. Now let's talk about how we get that information from the head up into the central nervous system and up to cortex. Okay, so same kind of overview slide laying out for you the steps of the pathway, and then we're going to break it down again piece by piece. So in this case now, the first order cell bodies are found in the ipsilateral trigeminal ganglion. And so that's illustrated here. We're focusing on the thicker line. Okay, the thin line, you're going to hear more about in a moment. So we're just focusing on the thick line here. Okay, so that's our tactile, proprioceptive inputs, cell bodies in the trigeminal ganglion. I mentioned here that some cell bodies are actually in the mesencephalic nucleus. You're going to hear about that a little bit later. Those are some that are doing some of the proprioceptive processing. Okay. So they come in via the trigeminal nerve. Right, so the sensory root of the trigeminal nerve. And so that's going to come in at the lateral pons. And they're going to synapse at the ipsilateral principal sensory nucleus. So that's located right there. And so you should note that that is basically around the same level where they come in, right? So as opposed to in the posterior column system, they come in and then they ascend all the way up to medulla before they find their second motor neuron. In this case, the second motor neuron is at approximately the level where they're coming in. All right, so we have our principal sensory nucleus here, and then the input, or the output of the principal sensory nucleus is going to, for most of the face and head, is going to go contralateral, so it's going to cross, and it's going to ascend in the contralateral ventral trigeminothalamic tract. Okay, there is a component that is going to ascend ipsilaterally that is involved in the oral cavity space. So we'll get to that and make that distinction for you. Okay, so then they're going to their third order cell bodies, which are going to be located in the VPL, which is just medial, as I showed you a minute ago, to the VPL. Then those axons, third order neurons, their axons are in the posterior limb of the internal capsule. In the internal capsule, it's arranged in a somatotopic way too, so that the lower extremities are more toward the retrolenticular aspect of the posterior limb, whereas the head is closer to the genital end of the posterior limb. And then they're going to terminate where their fourth order cell bodies are located in the contralateral postcentral gyrus in the most lateral position. Okay, so now let's kind of break that down. In terms of receptors, same kind of receptors, so we don't have to go through that again. But what you should recognize is that it's called the trigeminal nerve, right? Tricycle 3, right? So there's three kind of main branches of sensory input that's coming in. Okay, you have an ophthalmic division, that's V1, and so this kind of color-coded to help you see what part of the head that's conveying information from. Okay, then you have a maxillary division, and then you have a mandibular division. Okay, and so those inputs are coming in, and they are terminating in an inverted arrangement. Inverted, if you're looking at it from an anatomical point of view, in the principal sensory nucleus. Okay, so here's the V1 representation coming in from the principal sensory nucleus, and then V2 and V3. And so our general pathway there, right, is our peripheral afferents to the trigeminal ganglions where the cell bodies are found. They're going to terminate in the principal sensory nucleus that's in the pons. Okay, and so you guys know that's the trigeminal nerve right there. Okay. And so that principal sensory nucleus, so that you can see that in this particular view of the pons. Oh, I'm sorry, I forgot to flip it. So this one is an anatomical orientation. Principal sensory nucleus is located here, and the trigeminal motor nucleus is just medial to it. And so then over here in the drawing, you see the principal sensory nucleus and the trigeminal motor. So the blood supply is coming from the basilar artery. In this case, it's coming from those long circumferentials, the ones that reach way out around to get to the lateral positions. And there is also some blood supply that's coming from the superior cerebellar artery as well. Okay, so that principal sensory nucleus has two divisions to it. There is a dorsomedial division and then a ventrolateral division. Okay, and that dorsomedial division, that's the one that's getting the input that's coming from the oral cavity. And so it's important to distinguish it because those projections are going ipsilaterally to the VPL. Okay? Whereas the ventrolateral division, that's giving you all the rest of that external information about that, most of the head and face, is going to go contralateral to that VPL. Okay? And so they have two different pathways then that they're going to travel in, right? So the posterior trigeminal tract is carrying that ipsilateral input of the oral cavity, whereas the ventral trigeminal tract is carrying the rest of that information about the head and face. Okay, so that ventral trigeminal tract that's carrying that information from the contralateral principal sensory nucleus is found, it sits just, so if this is the if this is the medial lemniscus in here, the ventral trigeminal tract just sits right there. So it's just a little posterior in position to it, but they're right up against each other, okay, as they travel through. So here's medial lemniscus here in the midbrain, and so the ventral trigeminal tract is just right there beside it. Okay. Okay. And so again, that's heading up to the VPM. And so you can see here's the VPL that we looked at a moment ago, and then the head from that trigeminal input here in the VPL. And so you see that located there. So then again, here's ventral trigeminal tract is coming up. That's it there, going to the VPL. Okay, again, the representation of the head is located most laterally, and again, we have sort of this distortion based on the density of receptors with small receptor fields, high afferent innervation, and you have that in this perioral area, and so that's where you have the most fibers, most receptors with small receptive fields, most dense innervation. So the greatest area of cortical space occupied representing that. And so you have this huge perioral area of primary somatosensory cortex. Okay. And so I also want to mention about some proprioceptive input as well, that is coming from the muscles of mastication, so that is chewing, right? So moving your lower jaw up and down. Periodontal ligaments, that's telling you about your holding your teeth in place and that positioning. Their cell bodies are found in that mesencephalic nucleus. Okay, and so if you look at this little green line, the receptor's there and it's coming in, and you see the cell body, the first order neuron is actually there in the mesencephalic nucleus. And from the mesencephalic nucleus, you have projections that go to the trigeminal motor nucleus. And so that's good, right? Because that's the motor output for the muscles of the lower jaw for the chewing, right? And so that's going to help you regulate bite force. You have projections that are descending down that are going to go to the reticular formation, cerebellum, as well as projections that are going to the principal sensory nucleus. And so you're going to be able to coordinate things like chewing and swallowing. An example, this is when you know you've been thinking about a topic too long. So last night I was eating salad with my dinner. You know, I had a bunch of real nice baby arugula mixed in with some big, thick croutons. And I'm chewing that, and then I'm thinking about this. And I'm like, oh, that's a great example of this. Right? Because, you know, what was going on in terms of regulating how I was going to chew that. I had this super soft baby arugula, and then I had those big, hard croutons that took a little more crunching, you know, in order to break down, right? I had to regulate that bite force. And so while we're eating the salad with our dinner, my daughter goes and bites her tongue. You know, she's like, you know, it hurts really bad, right? You know, and I was like, just slow down, be careful. But she was missing that coordination component, right? She, you know, because we're trying to, obviously when we're coordinating our chewing and swallowing, we try not to bite our lips, tongues, cheeks when we're doing that, but sometimes things go a little wrong, right? So this just illustrates to you how this works, why it matters. Next time you're eating a salad or something that's got a nut mixed in with a nice soft something, you see the difference in what you have to do to actually chew that appropriately. Okay, so in terms of key differences then between the posterior column medial lemniscus system and the trigeminal thalamic system, what are the differences in these pathways? Okay, so you should know the differences in terms of where the first order neurons located, right? Are they in the posterior root ganglion or the trigeminal ganglion? What's the distance between the first order neuron and the second order neurons, right? So in that posterior column medial lemniscus system, for that long ascending pathway, right, there can be a long distance till you get to the second order nuclei. There's a couple of different second order nuclei for the posterior column, right? Nucleus gracilis, nucleus cuneatus, but you just have the principal sensory nucleus as that primary second order nucleus in the trigeminal tract. Where does crossing happen? Right? So crossing happens at the sensory decussation in the caudal medulla for the PC-ML system, whereas crossing is going to happen there in that pons level for the trigeminal thalamic system. Okay. What's the ascending pathway, right? So Is it the medial lemniscus, or is it the ventral trigeminal tract, or even the dorsal trigeminal tract, depending on what part you're talking about. Thalamic nuclei, right? They're different thalamic nuclei, VPL versus VPM. And then lastly, where's that somatotopic representation in cortex? So make sure you know and you can keep all of that straight. I want to give you a preview of thinking about the difference between the PCML system and the anterior lateral system that you're going to hear about on Monday. Okay, and so this is the PCML system drawn here. I'll try to give you the levels: cord, medulla, pons, membrane, and thalamus. And we're ignoring the green because that's the trigeminal, and we don't want to compare it right now. Okay, and so what are the differences here? This is really important in terms of, okay, where the first order nuclei are located, same place. Right, so that's good. But then, where does the crossing happen? Right, and where are the second order neurons found? Okay, that's very, very different between the anterolateral system and the PCML system. Okay, so the second order neurons in PCML, you know now, are here in the caudal medulla, right? And then the crossing happens there at the sensory zencisation. In the anterolateral system, the second order neurons are found there in the posterior horn, right? So that's found right there at the, around the level where they enter into the cord. Okay? And we're going to synapse there in the posterior horn. And so then it's the neurons of the posterior horn whose axons will cross at around the level of where they are. You can go up a couple. And they're going to cross there. And then it's going to ascend in the cord on the contralateral side. Okay, so that's important because now you've got touch and proprioceptive information ascending on one side of the cord, and you've got the pain and temperature on the opposite side of the cord for the same side of the body. Also, you should notice that the ALS system, there's a lot of branches coming off here, right? So there's collaterals you see here in the pons and in the midbrain, going to some different thalamic nuclei. Because in the anterolateral system, not only do you need to be able to localize the site of the pain, but there's this whole affective component of pain. That's sort of that emotional processing component of pain. There's a whole descending pain control pathway that gets input about the pain so that it descends back down to try to modulate that or reduce that fire. So those are some of the big differences between those two pathways. All right, so let's go back then to our mystery pathology. Okay, so we've got this 37-year-old woman. She's got tingling and numbness in her arms, legs, and trunk. Okay, so what's that tell you? Does that tell you anything about the level?
Yeah, it's about T5.
Yeah, so it's definitely got to be above T5 because I've got to get my arms too, right? So I've got to be cervical level, really, in order to have all of those areas. So it's got to be at least there, right, in order to impact both the arms and the legs and the trunk. She complained of clumsiness when walking quickly or up the stairs. What is that talking about?
No position sense.
So she's got proprioceptive issues. She's not getting the right kind of feedback to be coordinated in her movements, right? She feels like she has decreased dexterity in her hands. Right? So that implies, that she can't manipulate things as well with her hands, or she's dropping things. So she doesn't have quite that fine motor controlling feedback. All right, so the exam was notable for dysmetria on finger to nose testing. So I don't know if you guys know what that is. That's like if I was to stand here, right, and working with my eyes closed, but can I hit my nose? Yeah, I actually hit the tip of my nose. So I don't have any problems. So I'm good with that. Tandem gait was unsteady. Okay, again, that's going to be what kind of issue? That's gonna be a proprioceptive problem. Dramatic Romberg sign was present. Okay, so that's where you have somebody stand with their feet together, right, and then have them close their eyes, right, and do they stay steady or do they start to fall over? Okay, and so again, what is that? What kind of information is that? That's proprioceptive information about posture and control of your body position in space. We got a profound loss of vibration sense. What was the receptor that was good for that? Yeah, the Pacinian corpuscles were good for that, right? So that's going to be a tactile kind of stimulus, and we've got that issue in both lower and upper extremities, although it's worse in the lower extremities, right? But again, pinprick and temperature sensation was intact. Okay, and so what is that telling me about? That's my interlateral system. Okay, so now can you make a more educated guess about where the issue might be? Somebody brave enough to...
The cervical, at the cervical level, there's a lesion in the posterior column, probably like a blood vessel.
Okay, so now we're going to say cervical level, we're going to say posterior column. What part of the posterior column? Whole thing? Just one part?
Both sides.
But yeah, so I'm going to have it impacting both fasciculus gracilis and fasciculus cuneatus, and I'm going to have it on both sides of the posterior column if I'm having issues with both left and right side arms and legs, right? So I just had that to take us through that we did that, so I need to talk about what it all means. So when an MRI was done, what they found was a small lesion. Looks small here in the picture, but obviously it had to be a pretty large area of the cord that was impacted. Right in here is where the lesion is. So that's a little bit lighter in color right there than the surrounding tissue in the cord. And so basically what was happening was there was that being lighter in color indicated that there was less myelin in that area. So there was something that was causing an inflammatory kind of response in that area. It was impacting the myelin. And most likely this was a first episode of multiple sclerosis. And this is also, that's the fact that it was a 37-year-old woman, that kind of fits in with that as well, because a lot of times MS onset will start at around that age bracket area. And while it's in both men and women, it is more common in women.